Hello, hello, everyone. Hopefully, you can hear me out there. Hi there, my name is Jason Langley, and I will be your host for today's webinar. We really appreciate you guys taking the time to join us. Hope you are finding ways to stay cool today and enjoy the next 45-ish minutes with us. Over the next 45 minutes, we're going to be walking through some of the science behind fingerprint drug testing and drug testing in general. We're going to focus on what the body does after it ingests drugs, and how fingerprint drug testing works at a very, very technical level. We're also going to explore what that means for organizations and how to manage your workplace drug risk at your business. Today, I am joined by two excellent speakers. First, we have Catherine Adams, Research and Development Manager at Intelligent Fingerprinting. Catherine, would you mind saying hello and telling us a little bit about yourself?
Yeah,
What are you going to
thanks.
be walking through?
Yeah, thanks, Jason. So I'm Catherine Adams, as Jason has mentioned. I'm the R&D manager here at IFP. Been here for 3 years, just over, and I'm in charge of doing all the research and development into our products and sweat testing.
Fantastic. We're also joined by James Robinson, National Account Manager at Intelligent Fingerprinting as well. James, feel free to introduce yourself. Tell us a little bit about what you're seeing in the workplace right now.
Yeah, thank you, Jason. Yes, good afternoon, everybody. It's a pleasure to join you this afternoon. Yeah, I've been with Intelligent Fingerprinting now for around a year. It's been a very busy year, and I spend most of my time speaking to health and safety professionals and HR professionals about their approach to drug testing in the workplace. So I gained lots of insight in that time, which hopefully I can share some of that with you guys this afternoon.
Perfect. Thank you both very, very much. So our agenda today, we're going to be examining, why the science matters. We're going to be covering some questions that many of you have encountered, and then we'll be moving on to outlining the science behind fingerprint drug testing. After that, we will be looking at different operations, the challenges that they face, and how a lot of businesses, a lot of clients of ours, uh, choose to leverage workplace drug testing at their sites. Finally, we will wrap everything up with a Q&A session where we'll answer all your questions. A little bit of housekeeping before we get this kicked off here. This session is being recorded, and we will make sure to share the link with you all afterwards. We have a Q&A that you can see. It might be in the bottom of your screens there. Feel free to pop in any questions that you have. We're going to try and address as many of those as we can during our Q&A segment. But if a question comes up later, or if we don't have time to go through it today, that'll be perfectly fine. Don't worry. One of us will make sure to reach out to you directly so that we can get your questions answered. With that, let's look at some of the most common questions that we hear. These are probably some that you've heard already, some that you've run into, some that you're thinking yourselves. A lot of businesses, for a lot of businesses, fingerprint drug testing is still quite a new concept, I think. Pretty safe to say. Now, I could answer all of these questions myself, but I wouldn't be doing them justice. Catherine and her team have put an enormous amount of work and science and engineering into the system. Catherine is going to take you through all of this stuff a lot more properly than I ever could. And then once she has, James is going to, tell you about what he's experienced and how that kind of ties into day-to-day operations. But the one thing that I'll say is if these questions are sitting at the top of your mind, you are exactly in the right place. I think that, we encourage people to ask questions like this because not only does it allow us to showcase the hard work that all of our team has been doing, but it really allows us to in real terms showcase what makes us different and how we can help businesses work a little bit better day to day. With that, I'm gonna stop talking here. We'll hand it over to Catherine, where she will break down the science behind fingerprint drug testing. Catherine?
Okay, um, first slide then, please. Okay, so basically today I'm going to go through the science behind it. I'm going to try and make it not too scientific and when I can explain what we mean by certain words. So the first thing everyone's wondering, right, is what happens to the drug after you've ingested it or inhaled it, whatever, however they've taken the drug. Well, the first thing that happens is it gets absorbed. So depending on how you've taken it, if you've eaten it, it goes into your stomach, which goes into your intestines, and it gets absorbed through your intestines. Or if you inhale it, like cannabis, for example, then it will go into the lungs and go through the bloodstream. So then the thing they all have in common is that it goes into the bloodstream. And then it will circulate around your body and it'll get to the liver. And the liver is a core critical organ, which then breaks down and metabolizes the drug. So then once it's metabolized and broken down, we then have these things called metabolites, which I'm going to explain in the next slide. But the point is the drug is now broken down and then it carries around your blood and eventually it will go into your urine. It'll end up in your oral fluid glands. You have two big glands here, which is how they can detect the drug in oral fluid. But then critical for our technology is it ends up going into your sweat. So if we go next slide, I'll try and go into more detail there. So basically, as I said, the drugs are processed in the liver and they get broken down into metabolites. And metabolites are just, if you think about a car with the fuel goes in, runs the engine, and then the exhaust fumes is the byproduct, right? And that's what basically metabolites are. They're a byproduct of a drug. So the parent drug has now been broken down into different metabolites. And not only does our test detect for the parent drug, it also detects for the metabolites. So it's not only just about the parent drug, it's about looking at like the trail that's left behind, because even though the parent could be broken down, we can still find the metabolites in your sample, in your sweat. And this is the same for all urine and oral fluid. They'll also be looking for metabolites as well as the parent drug. If we go next slide, please. So then the metabolites obviously circulate around your blood, as I just explained. And then for us, it passes in to your sweat glands. And what's interesting is that on your hands, you have eccrine sweat glands that are there. And it's a really high density packed area compared to anywhere else on your body. So it's not only on your hands, but you also have eccrine glands on your feet as well. So this is a great place for us to sample, right? It's not invasive, it's not in a strange place to collect because you can just give your hands and give your sample. So what happens is the sweat then gets released by the eccrine glands. So not only does it release water and salts like we all know, right? You know, when you're sweating and you accidentally lick your skin, it's salty, right? You'll also be letting the drug and the metabolites of the drug come through the sweat gland as well. And that's what the beauty of our technology is. It harnesses that normal process that happens in the exocrine system, and we've then harnessed the capability and created a product which then allows us to detect those drugs and its metabolite in sweat. So if we go next slide, please. Okay, so then why use fingertips? Well, it's easy to collect, isn't it? You can give me a sample in front of me. I haven't got to worry about you going to the toilet or me being hydrated properly to be able to pass urine. I'm not going to put something in your mouth to collect a sample. It's literally you giving me 10 fingerprints. So each finger is given for 5 seconds. You press onto a sample pad and that's it. It's dead easy. It's truly non-invasive, doesn't hurt. The other reason, again, is what I've just went to explain before, is the density aspect. There are loads of sweat glands on your hands. And if you think about it, you know, when you get nervous and you start sweating from your hands, right? That's the reason why, because it's such a highly density, like packed area, you get the natural sweat on your hands. Even when you think about when you touch a cold glass and you leave a print behind. Think about CSI and forensics, right? There's always prints left behind. But the one beauty about our test is that we have no biometric information of you at all. Because if you think about it, you're giving me 10 overlaid fingerprints and I can't now decide between it. But the point being, the material we use is a porous material. We don't ever look for any ridges or anything that you see on CSI. It's just purely getting the sweat, which then has the drugs collected onto the sample pad. So there's no ever link back to the person for our technology. If you go next slide. Okay, so how the fingerprint test works. I think this is really cool because it's truly point of care, doesn't hurt. So we collect your your sweat samples as presented. So you walk in and the person who's doing the drug testing, they'll say, can you give me 10 fingerprints? So you pass them the cartridge, you then collect your sample on. Again, each finger's pressed on for 5 seconds each. So you have 10 prints now, which is a really good sample size. So there's enough drug there that will be deposited if you have taken drugs. And then the sweat's now absorbed, if you like, onto the sample pad and it's there present. And then what happens is we have an extraction buffer, which is inside a cap or a buffer cap, sorry, that's on the lateral flow cartridge. And all they have to do is slide the tamper evidence seal down to protect the sample so there's no contamination or no one could accidentally put another sample on. It's truly to that person, you're watching them do it. And then you burst the buffer clip. And then that allows our extraction buffer, which we've made, to extract the drugs from the sweat sample. And it runs like a normal lateral flow. So remember when we had COVID, not that everyone wants to remember that, but it's the same basis, right? The fluid will migrate up, what we call nitrocellulose, which is a membrane. And then on there, we have 4 different test lines that detect 4 different drug groups. So opiates, methamphetamines, cocaine and cannabis. And what happens is, because of our technology, it's fluorescence based. So you with your naked eye can't see anything. And compared to a COVID test, remember how you could see the lines? That's a visual test. So with our test, we need a reader, which is what we have a DSR Plus for. And when we use that reader, it's truly unbiased, right? There's no us visually looking like, oh, is there a line? Is there not? I'm sure you all had that instance when you were doing your COVID test before, but with our technology, it's fluorescence and the reader is told a certain ratio. And if you're above or below that, it dictates what the outcome is. So if you're above the ratio, because it's a competitive assay, you are negative. And if you're below it, you are non-negative. Now, the reason why we say non-negative, like any drug screening test, not only just in sweat, but urine and oral fluid, it's a screen. We need to get the final confirmation from an LCMS lab. So if we go, I can't remember next slide if we go into more detail here. Yeah, so then when we're doing the screening cartridge capture, we use antibodies and they've got the fluorescent marker on. Now, the antibodies are like a lock and key theory. That's the easy one to explain. So the antibody is the lock and the drug is the key. So if you have the drug in your sample, then it will fit into the lock perfectly. So imagine if somebody had taken cocaine, then our cocaine antibody will find the cocaine within the sample. And then what happens is it migrates up the strip, and depending on if it's present or not, it will either bind to the test lines. So if there is no drug present in your sample, then the antibodies will bind to the drug line, which is similar to other tests, especially like urine and oral fluid. They're still competitive in nature. So if we go to the next slide. So like I've just explained, the reader reports are negative and non-negative on the screen, and that's based off a screening cutoff that we've given and validated. If you're non-negative, then the subject is asked to go wash their hands and come back and collect a fresh sample of sweat, not touching anything. And the reason why this is really good is because it removes any issues or any like feedback back, oh, like I touched a door handle, somebody might be on cocaine or something like that. It removes everything and it's truly washing their hands and coming back and collecting a fresh set of samples. These samples, we collect 2 back to back, and they get sent to the lab and the LCMS gets ran on them and they come back with the final result and the final verdict. So it's either positive or, well, hopefully positive and never negative. If we go to the next slide. So the common myths and scientific facts like that we always get asked is, again, it stores my fingerprint. No, it doesn't. As I just explained before, it's overlapping of samples. I can never tell who it is. You think about CSI and all the crime things you see, and they're dusting for prints. It's only a solid, good print that's always good, right? So we never collect any fingerprint sample type. We can't ever tell who it is. I touched a contaminated surface. We've set our cutoffs deliberately to account for that. Like, we understand, especially because Professor Mel Bailey, she wrote and did some research at the University of Surrey about contamination in the workplace or in general, and she found a high level of cocaine on like banknotes and surfaces. So we've already taken that into consideration when we've set our cutoff for our screening cartridges, and that's why it's so important when you are non-negative on the screen that the hand wash happens and they don't touch anything, because then If they give that print again and they're still positive on LCMS, then we know it definitely has come and been excreted from themselves. I don't sweat. Well, the funny thing is, even if you don't sweat, you're definitely going to be leaving a trail. So even the tiniest amount of sweat will still give us a decent amount of drug level in there. Um, and it's still the collecting 10 prints also increases the sample size too. A small sample means less accuracy. Not necessarily. In our case, it doesn't. Like that's why we collect the 10 prints over time to try and rule out the don't sweat bit and the small sample. Even it's the quantity of drug that's there rather than the concentration. And if you go to the
Awesome.
next slide, is that it now?
Yeah, yeah. Well,
Okay,
yeah,
sweet. There we
yeah.
go.
The one thing there was one question that that's come up a few times that I've heard a few times as well. You were talking about so someone does their screening test, right?
Yeah.
And then if it comes up non-negative, at
Yeah.
that time you mentioned that they have to wash their hands after that, right?
Yeah.
Is is there some reason that they shouldn't just wash their hands before or to get a clean sample or
Um, not really. We've validated to just be as presented, so the cut-offs are set appropriately to deal with the contamination within the workplace. And that's why then the hand washing in the second time is then 100% necessary to collect a fresh sample within a 15-minute window.
And can I just ask one more question to confirm? So you said that our system looks for both the metabolites and the parent drug. Is that
Yeah,
right?
yeah.
So both can be, leading someone to showing some kind of indication of what's the word that I'm looking for, James? Some kind of indication of impairment or something. Is that right?
Quite a
Yeah.
significant concern. Yeah.
Hmm. Excellent. Well, yeah, thank you so much. Thank you so, so much, Catherine. Yeah, such super valuable insight. I really, really appreciate, you taking the time and making those things nice and simple for us to understand too. It's wonderful. With that, I'm gonna hand it off to James. James, can you tell us a little bit about what this means operationally for businesses? What are you
Yeah.
seeing in the day to day?
Yeah, absolutely. Thank you, Jason. And thank you, Catherine, for that really, really good overview on the science behind it all. And actually, one thing that's worth mentioning here is that this entire solution is based on science, and science is empirical, right? So science is based on fact and inarguable fact, and any solution that's based on empirical research and evidence is a trustworthy solution. And I think that's one thing that speaking to business owners and decision makers in the organizations with whom we work is that trust is imperative. We're very aware that the results of a drug test have quite far-reaching and potentially grave ramifications for both the individual employee and for the company itself. And whilst the explicit understanding of the science behind drug testing is probably out of reach for the vast majority of business professionals and business owners and decision makers, I think that it's really important and actually really a prerequisite that before you deploy any kind of drug testing solution, that you understand that that particular that organization that's providing that solution has done that kind of that research and is has science that underpins the solution that you're using. So I think that moves on to the next slide there, Jason, in that really what we need to do as part of from a business approach is you need to assess that company as opposed to the actual literal science that's within that company. So how do you do that? Well, there's a few things that we advise our prospects and our clients to do when deciding how best to approach drug testing in the workplace. And the first thing is really to see the accreditations of that company. And specifically the ISO 13485 accreditation, which is really focuses on the efficacy and accuracy of medical devices. That really is a third party affirmation from an independent body that gives you that confidence and faith that the technology you're using is appropriate to be used in your workplace. The other thing really is to look at the current clients of that particular organization. check to see if your peers in your industry are using exactly this kind of technology. That's obviously something that can affirm that you're dealing with the right people who have already helped and supported your peers throughout the drug testing process. The other thing is to look at historical results. See if those things are the results that are coming through are kind of in line with what you're expecting and in line with what they're saying is likely to occur. And then primarily, I think above all of it is build a relationship with these people, build a relationship with the people that you're effectively placing a great deal of trust in a very delicate process within your health and safety function and within your HR function. So really, they're the key kind of elements for assessing whether that's something that's going to be an appropriate solution for you. Thank you, Jason. I think the next slide will be a good one. So yeah, so what do businesses care about, and actually, what are they looking for, and how does the science dictate where you want to go with your drug testing? So what I'm seeing when I speak to professionals at the moment is that there is a significant incline, a significant rise in solutions that focus, or the desire to implement solutions that focus on recent drug use. So there are certain organizations that really want to look at historical drug use. There's some people where it's an absolutely hardline, zero tolerance approach to drug use. And what I mean by that is that some people want to know that if an individual or an employee has ever used drugs at all within their employment, then they don't want them working with them. Certainly for that, you're going to be looking at a long period of drug use. And for that, there are other things that are driven by science that would determine that for you. Hair, for example, has a very long period within which drug use can be detected. But as I mentioned, the vast majority of people we speak to really want to focus on fitness for duty, how potentially impaired people may be on the day. And that's where you need to look at the science, which will dictate what method you use. So again, referring back to Catherine and the different systems in the body, metabolize drugs in different rates. And the excrete system, which is all around sweat, metabolizes that quite quickly. So therefore that gives a very short indication of recent use. And also, people want it to be non-invasive. We are in 2026. We're not in the 80s anymore. We don't have to be handling people's urine. We don't have to be squeezing out saliva cubes. And actually, that experience, the employee experience, is very important to people that I speak to on a regular basis. In fact, just the other day, I was at a port skills and safety event in London, speaking to a couple of our clients who recommended some of their peers from another organisation who were absolutely sick and tired of dealing with other employees' urine. That's just a primary driver for them, is that they wanted to have that experience both for the individuals who are delivering their samples and the individuals who are testing those samples. It's obviously going to be the key thing there is to have that less invasive kind of approach. Okay, next slide there, Jason. Thank you. So yes, so this is really important. This harks back to what I was saying earlier. So what are you trying to identify? Are you trying to identify whether your employee is fit for duty, or are you trying to identify whether they have historical drug use in their past? And there is no perfect drug test. Every test, as Catherine mentioned earlier, has a certain level of accuracy, and actually it's the same across the board. So all drug testing have somewhere in between a 95 and 97% accuracy level. You have, and again, that's due to the science around the collection and the analysis of these samples. That means that you do have a 3 to 5% window of doubt where that could be a false positive. So it's absolutely crucial to have an easy path to confirming that sample with the LCMS that Catherine mentioned earlier in her presentation, to almost upgrade that non-negative result into a positive result. And when it's a positive result, that's a legally defensible result. And from then you can take appropriate action from that point onwards where you really couldn't when it's just from that initial screen. So it's really the only right test is the one that fits your policy. It's vitally important to match your policy with your testing. And so you need to find the right test, and often it's science that will guide you to that right test that fits your policy. Okay,
Perfect. Thank
next one,
you so
Jason.
much for
That's
that,
it.
James. Yeah, yeah, I really, really appreciate that. I think it's a good thing to be able to tie some of this theory into, real life operations and see how this affects businesses day to day. With that, we can move on to our Q&A session here. We've had quite a few questions come through. Feel free to keep on popping your questions in here. We've had a few pre-submitted. I think, James, you kind of talked about it already, but I'll ask it one more time. How accurate are fingerprint drug tests compared to urine or saliva? Are fingerprints more or less accurate? What's the percentage? I think if you wouldn't mind just.
Yeah, absolutely. I can reiterate that for you, Jason, and thank you, whoever that was who asked that question. And yeah, the very simple answer is that there's a par. You're on a par with all of the other kinds of drug testing. So yes, absolutely. We obviously have a view of all of the results, and yeah, absolutely. From the results that we can see from non-negatives across all of our clients, then it really rings true with what I've just said. You have between a 3 to 5% window of potential false positives come through, and that is in line also with saliva and urine testing. So it's a well-established fact that across the board you have that slight small window of doubt, but between 95 and 97% accurate is rings true with our results.
Perfect. There's two questions here that I'm going to fold into one. So, what about medical marijuana? Does your test for legally obtained cannabis? How can we support where a colleague has been prescribed medicated cannabis to support long-term ill health conditions?
Yeah, well, I can certainly cover that one off for you guys if that's okay. So yeah, it's an interesting one. Medical cannabis is becoming more and more accepted as a regulator for pain relief. It certainly has those analgesic qualities that can be beneficial for some people with chronic pain. Um, our solution, again, we hark back to the fact that it's science, right? So it's a science-based solution, and a science-based solution doesn't know the circumstances around which that person has ingested that drug. It just recognizes the psychoactive compound that's in cannabis, which is THC. So really what would dictate what happens when THC is recognized and picked up in a test is really down to the policy behind that testing. So if your policy declares that you have an obligation to declare any kind of medication that could have an impairment factor. So think also codeine, cocodamol, and also medical marijuana, medical cannabis. Then you have an obligation to do that. There's really a perspective on this could be that actually there is somewhat of a stigma still, I believe, with medical marijuana, just as there may have been a stigma with codeine in the past. But they're both impairing substances, both impairing drugs. So really, it's a question down to your policy whether that declaration of prescription of medical marijuana is acceptable, just as a prescription for codeine may be acceptable. But our solution won't know the difference between the two.
Excellent. Thank you, James. Do you have any thoughts, Catherine? Or like has James kind of covered everything there?
Yeah, it's more a fact of saying that it's present, not necessarily like exactly like are they taking their dose, or it's just
Yeah.
about whether if it's there in their sweat or not. That's not really for us to say about how you handle that person. Again, it's like Jane said, it's linked to your policy within your business, really.
Perfect. Next question here. Are there methods developed yet to beat the system or that can give a false positive? The person here says, like mouthwashes can affect saliva tests.
Hmm.
Some drinks can give positive. I think that they mean urine test here. I think, the one thing that I have heard and seen is like a simple YouTube video search will come up with ways to cheat urine tests. Are there ways for people to cheat our test? Like James, Catherine, are
Um,
there any ideas
So
that you
yeah,
have?
so look,
Not to encourage bad behavior, but
no, of course, of course, we're giving away some trade secrets here. No, look, maybe Catherine can cover off the scientific side of this. I speak about this pretty much every time that I kind of go through a demonstration of our solution with prospective future clients of ours. And for my mind, the only really way that you can cheat this is if you do what I call the hover job, which is where you don't actually press on the on the collection plate. And there is best practices that we can advise our clients to ensure that that doesn't happen. It's actually quite obvious when somebody's doing that. But to my knowledge, apart from that physical kind of detachment of your finger on the collection plate, scientifically, I don't know of any. But we have heard some hilarious stories of how other people have cheated kind of other methods of drug testing. If anybody wants to hear them, feel free to get in touch with me and I can talk you through some of those. But to my knowledge, with ours, I don't know of any.
would it be correct to say that the person being tested does not need to wash their hands before being tested the first time?
yeah, that's correct. Yeah, they can just walk in as presented and give a sample.
Excellent. Um, do we have any more studies which consider contamination, or perhaps can you talk a little bit about more about the surface contamination that you were mentioning previously,
Yes.
Catherine?
So obviously our way we collect is very different to other urine or fluid tests, right? They have very different contaminants, especially with oral fluid. You think about everything you eat. When they run their validation studies to confirm their test work, they have to think about all those aspects. And with us, it's similar but different, right? So think about everything you touch, handle, apply. So when we do our validation for our products, we use all those things. So we apply things to hands, we then give a sample and we see if it interacts and interferes or not. So those things are already included in the validation. And so we already have like mitigation and that's the cutoffs are set the way they are, which is linked to like the contamination with the background of the drug, but then removing the issues of any interference from other things like, I don't know, hand cream or sun cream or even remnants. Like I know we've discussed about how cocaine's quite rife in our society. So like if that's present, then that's all been accounted for. And that's why the hand wash again is really crucial for the confirmation step.
Perfect. Next question I have here. When a non-negative result is determined and the person being tested has washed their hands prior to
Yeah.
giving the second test, would I send both test cartridges in for lab analysis or only the second test that has
Yeah.
not had the solution activated? So I think that, yeah, you can probably talk the second cartridge is actually a little bit different, isn't it?
Yeah. So the first cartridge has the buffer burst on, and that's used with the like the reader, sorry, DSR plus. And then once that's done, you just discarded that cartridge. And then when they go wash their hands and wait 15 minutes, it's a different type of collection. So it's the same look of it, but instead it's like a black material that we collect the prints on. And you collect two, so we've got an A and a B sample. And the second sample that's collected will be ran in the LCMS. And the first sample will be kept back in case you want to challenge it or anything like that happens. So there's always two collected, but the second one's always gone run through the LCMS with the result given. So you'd send two, but it'd be confirmation, which is very different the way they look.
Yeah, the way I explain that is it's the secondary the cartridges that we use to take that secondary sample are effectively transportation devices.
Yeah.
They're effectively a method by which we can collect that sample and send the sample back to over to our lab partners to run through the LCMS.
Perfect. Next here, are you able to understand the levels of drugs within the samples given? Secondly, if someone is prescribed a medication at a certain dose, but it comes back non-negative for that medication, yet a higher dose prescribed. So I think that what this person is asking is, are you able to tell the levels of any certain drug within the screening test? Catherine, you might be the best to.
Yeah, so in terms of our screening, like cutoff, that's assessed based on clinicals that are ran on like dosing of drugs that are safe to give in a clinical setting, right? So that's the only level we can tell, and like it's a qualitative test, which is similar, the same to all matrices, because urine and oral fluid, majority of them, it's yes or no, right? If you can see a line, if you can't. So it's the same way here. We set our cutoffs, which in the screening device, which is in picograms. So we're very small because think about it, the sample's really small too. But we're able to detect in picograms in a 10-minute test, which I think's amazing for a lateral
Yeah.
flow device. We can get down to that picogram level. So we can't give you explicitly in the screening device how much is there. But then when you go for LCMS, that machine, it's this lab analyzer, right? It's much more accurate and can define exactly how much is there. And then again, that's worked off a cutoff that's set on the LCMS, and they'll say if it's above or below the figure they've got back compared to the cutoff. So the answer is no, but if you screen positive, then you must have taken more than what the cutoff stated. And the cutoffs are stated in the IFU as well, for the device, for the different drugs.
Excellent. Next question here. Does this also pick up alcohol?
No, but there is a byproduct with cocaine. Is it cocoethylene? We definitely detect that on the LCMS. So that does cross-react to some level, but we don't do alcohol per se. It's normally like how it interacts with the drug, and if there's a byproduct that's made because alcohol's present. It's not like a breathalyzer. It's
Yeah.
linked to the interaction between alcohol and the drug in your body, and it's specifically for cocaine.
Can just add on to that that if an organisation is wanting to assess their employees to see if they have drunk alcohol, we do provide type approved breathalyzers with a from the Intoximeter brand that allows you to screen in tandem with the drug. So it's a modular solution from us. So you have the real innovation, which is the drug testing and the technology that we've been talking about up to this point. And then you have the secondary module, which is the breathalyzer style way of indicating the presence of alcohol within their employees. So yeah, so we have a holistic solution for drugs and alcohol. The real innovation in our company, though, is within the drug testing.
Perfect. Another question here. Has a positive result with this technology been challenged legally?
Hmm.
I think, James, you might be like you work with quite a few different clients. Have you heard
Yes,
anything about the legal
yes.
defensibility of all of this?
So look, so to my knowledge, something about which we're really quite proud, to our knowledge, that we've not had a client that's been taken to tribunal or court based on the results from our technology. Because of the level of science and because of the depth of the research and the thought and the engineering behind our solution, primarily from Catherine's team, there are various different fail safes within the reader, where if there was a challengeable situation that occurs, I think of maybe, for example, an out of date calibration or an out of date cartridge, something along these lines. There are fail safes within the system that would effectively stop the test as it goes. We would rather you not test than test in a way that can be legally challenged. And I think that's really important to kind of think of that. And we've developed this solution from the ground up, right? So we literally from a blank page with an extensive knowledge of how people can be challenged in the drug testing arena. So we've got the technical fail safes within the solution, but also process. We've got, we're very, very stringent with the process, and our training team are fantastic in guiding our clients through the process of recording the right information in the right time, in the right places, and taking the right precautions and the right actions to prevent any kind of challengeable situation. So in short, no, I don't believe we have had any kind of legal challenge on the strength of our solution.
Yeah, the chain of custody is quite strong, isn't it, when they collect the sample?
Very, yeah,
It's
yeah,
very
absolutely.
easily written as well. So if you follow the
Mm.
guidance and the IFUs and the consent pad, because even the consent pad tells you like what to do
Yeah,
or what you shouldn't do,
exactly.
then it
I
helps with
think
that legal bit.
yeah, and I think the key thing there, the one thing that's going to lead to challenge is ambiguity, right? So, and ambiguity and interpretation. As soon as you start interpreting anything, then there's an element of perspective and there's an element of opinion that gets involved in an interpretation. Even if it's a case of writing down the results from the reader onto a bit of paperwork, there's an interpretation there. A really good example there is the strength of a line. You know, that's an interpretation. If there's not a binary result given to you and it's just a line, there's instantly a decision that needs to be made. Is there a line there or not? Is that my buddy? Do I not like them? That could potentially
Yeah,
determine whether that line is there or not. And that could be challenged. With our solution, the results come to you in a binary way. So it's yay or nay. It's either negative or it's non-negative. And when you have that clarity, that takes that interpretation off the table, then obviously you've got a much stronger kind of chain of custody in that way,
yeah,
and less to challenge. And that's a great example actually of the small incremental improvements in the process and the small incremental focuses on the chain of custody, by which, by the way, a lot of people I speak to, when they talk of the chain of custody, they think of the chain of custody from a non-negative onwards. We actually consider the chain of custody from the minute they sit down to do their tests onwards. So to have that robust process and have those incremental gains throughout those processes means that it's a really robust solution that's very, very difficult to be challenged.
yeah. And the also beauty about having the reader system, which I guess is different. And one of the benefits compared to urine or fluid is that we have the reader which stores the results, and there's no handwriting. You can have the printers included. You can print the label, which then stick onto the consent. So in terms of the traceability aspect, there's no yeah, ambiguity, and even you're supposed to have the same, ID you've put onto the reader will match what's on the label. So in terms of that traceability element, it's all there to protect you and the donor to make sure it's all the right sample, it's the right paperwork.
Perfect. Another question here. We have loads of questions coming in, guys. How long do fingerprint samples that are sent to the lab analysis last, or how long are they viable? Saliva samples degrade over a short period of time, and in my experience of testing, are fingerprint samples more viable, or do they last for longer?
The lab will hold your samples for up to 3 months. So if you wanted to challenge or anything happened, you want to sample the sample, they'll keep it there for 3 months, and then they'll do analysis on it and give you the result back. So that's all done via our UCAS accredited lab. They have the method to show the stability of the samples.
Excellent. Another question here. I understand the this idea of batch testing can occur. So is chain of custody lost? Maybe James, can you tell me a little bit about how chain of custody works when you're doing batch testing?
Yeah, absolutely. So yes, the batch testing function is a really unique and a very efficient way of running tests, especially random program tests. So just for those that don't know, there's a function within the DSR Plus reader that basically allows you to run tests in a very quick way. So the primary thing that delays the time to result is the development of the cartridge. And again, if you just think back to your COVID days, you had to wait 15 minutes, right, before you could read the result, before it give you a viable result. Well, in the same way, it's not quite as long as 15 minutes. I believe it's 10 minutes that we have to develop our cartridges. So that's the main hold up to getting a result. So if you were to everybody's give their sample in the same time, and then activate that buffer clip, that means that you know that that batch of up to 6 cartridges are developing at that same time. You can then effectively choose a different mode on the reader, which will then straight away tell the reader not to wait for those 10 minutes because you've got ready samples to be analysed straight away. Now, that is a very separate aspect from the chain of custody. So the chain of custody there is ensuring that you've got all the right information from the donors and you've got that all in the right places and you have their consent and everything, all the boxes are ticked pre collection of the sample. So that won't be affected because you do all of that in a preliminary way before you take those samples. With regards to coming across a non-negative result when you go through a batch test function, then that's just a question of having somebody ready to take that individual away and carry on with the back to lab process whilst you continue testing the remaining screening tests in that batch test batch, if you see what I mean. So yeah, so the batch testing will not, and we have clients who use batch tests on a very, very regular basis, and they've never had any kind of issue with the chain of custody whilst using that technique, that method of testing.
Yeah, the critical thing is just labeling your cartridges, which has helped with the consent pad. I'm sure that gives you a number, doesn't it? And then you can relate the number to the person. So just
Yes,
ensuring like
exactly.
there's a label on the back of the back of the cartridge,
Yeah,
which you can write the time and date, which is what the IFU explains to you. Like when the sample's collected, you take it from the donor and you write their ID, whatever you're going to assign, and then with the date and time. Is a neat trick if anybody on the call is already using our technology. You can write on the buffer clip. That's what we do in R&D. So you can write just any whatever number you want, so you can see it as you put into the reader, it's still outside the reader. That's what a neat trick we use. Rather than remember, oh no, what do we
excellent.
put in there? You could always just write something on the buffer clip as a prompt. That's always a neat trick.
Love that. Pro tips from the cat from Catherine. Love it.
Yeah.
Perfect. So we're starting to run out of time, so I'm going to ask one more question here, and if we can just try to keep it a little bit short, I just, want to be mindful of everyone's time on the call here. Um, so one question is, can you please compare the window of detection between urine drug testing and fingerprint drug testing?
Yeah,
Okay.
absolutely. I can start fielding that one for sure. So the window of detection for urine is depends on the drug that's going to be detected. THC, which is the psychoactive compound in cannabis, famously stays in your urinary system for a long time. So up to 30 days it is a reasonable thing to say about THC in your urine. And that obviously has its own, if you're looking for fitness of duty, if you're looking at potential impairment, that comes with its own challenges because you just don't know when in those 30 days that THC has been ingested into that person within that sample. With our solution with fingertip sweat, due to the difference in the systems that I mentioned earlier, so with the excrete system, we can detect THC that's being ingested within 24 hours of that test. So the window of detection there is 24 hours, which clearly gives you a much greater suggestion of impairment.
Perfect. Thank you very much. Sorry again for dropping earlier, everyone. Um, it's, happens, I guess. Well, with that, I just want to thank everyone who's joined today. Really, a big thank you to Catherine and James for all of your insights. Before we close, I think that there's a few key takeaways that we can keep in mind. Number one, fingerprint drug testing, from everything that Catherine has said, is a scientifically valid and verified system that many organizations are using already quite successfully, from what James has been saying. It detects drugs using very non-invasive and very technically impressive methods. Also, we would really suggest, like James was saying, look at, what you want to test. Start with the why before you look at the how, right? Look at what your objectives are and then figure out how you want to go after those objectives afterwards, right? With that, we'll bring today's session to an end. Thank you all for being a part of this today, and we look forward to continuing the conversation. Please feel free to reach out if you want any more information, any more guidance. If you want to know a little bit more about the science behind all of this, we're very, very happy to talk you through it. Hope you have a wonderful rest of the day. Please stay cool, and we look forward to chatting to you soon. Bye now.
Thank you. Bye.
Bye bye.